Hi everyone, welcome back to another episode of the Metabolic Link. I'm your host, Dr. Angela Poff. On today's episode, we have an outstanding presentation from Dr. JEFF VOLEK entitled Exploring the Keto-Adapted Phenotype, Focus on Muscle.
Identified three areas that I think are underappreciated in terms of how ketones and ketogenic diets impact muscle. One of those is related to glycogen metabolism and some interesting data that we published close to a decade ago, I guess, but really hasn't been followed up on to my knowledge. Another one is related to the idea of ketone production in muscle, particularly skeletal muscle. And then I want to share some really provocative newer data that has to do with ketone metabolism and heart function, cardiac function. You know, really, it's almost the world turned upside down when you look at the new evidence around ketones that's emerging on a weekly, monthly basis. Ketones are being described as super fuels now, and longevity metabolites, and a superior energy supply. So, it's really a fascinating time right now to be rehabilitating this negative connotation around ketones and embracing all this really exciting science around ketones as a beneficial molecule. So following up on that, I really think it's important to understand ketone concentrations. And one little fact that's fascinating is that ketones span over 4 orders of magnitude in the circulation, depending on the stimuli and the type of diet and conditions you may be experiencing. And so the vast majority of people on a standard American diet are, their ketone levels are probably between 0.1 and 0.2, maybe, occasionally in the morning after an overnight fast up to 0.3. And when you restrict carbohydrates and adopt a ketogenic diet, those levels increase by an order of magnitude. And this is not a linear scale, by the way. This is a logarithmic scale. So they'll go up, and we kind of define this as nutritional ketosis, somewhere between around 0.4, 0.5 at the lower end, all the way up to about 4 or 5 millimolar. And of course, we know we can achieve this too with exogenous ketones now, at least transiently. So this is quite a bit higher than what most people are experiencing on a typical American diet. And then there is a thing called starvation ketosis, and you can bump levels up a little higher than you can with the ketogenic diet. So you might see levels 6 to 7, 8, 9 millimolar, but that's still quite a bit different than what you experience in ketoacidosis, which of course is very serious, life-threatening, dangerous situation. So understanding, where your ketones are in this range is really important. And I might, go as far to propose some different nomenclature around this, because, this idea of what is normal, what is the normal ketone level, or maybe a better question is what is the optimal ketone level for an individual or for a particular therapeutic target. These are really important questions, and I think a good argument can be made that this level of ketones in the range of nutritional ketosis is an optimal range for many people. And so terminology we might consider is that a normal range of ketones is euketonemia, is this 0.5 to 4 or 5 millimolar. Much like with glucose, we have hypoglycemia, euglycemia, and hyperglycemia. So if you're on a typical American diet, you have hypoketonemia, and then reserve hyperketonemia is something that's more along the lines of ketoacidosis. So there are many ways to elevate your ketones, and traditionally we've thought about ketogenic diets as a standard way to do that. We now have a variety of exogenous ketone formulas that range from salts to free acid to esters, and those clearly acutely raise ketones transiently. But if you use those repeatedly, you can keep your ketones levels quite elevated over time. Fasting will raise ketones because they're naturally low in carbohydrate. Exercise transiently, you get a bump in ketones. Experimentally, there are studies where you can infuse beta-hydroxybutyrate salts and induce acute levels of ketones. I'll actually show some data in terms of cardiac function later. And interestingly, if some drugs also induce ketosis, like the SGLT2 inhibitors that result in mild ketosis. And this idea of keto adaptation, I really think that should be reserved for the set of adaptations that occur to a ketogenic diet or carb restriction, because I don't think you're going to get the same adaptations from taking exogenous ketones repeatedly and still consuming carbohydrates. There's just a fundamental difference in terms of some of the underlying physiological responses and adaptations that occur. But that takes time. We're just beginning to unravel some of the various adaptations that occur to ketogenic diets and various forms of elevating ketones. Incredible science going on with a ketone-fueled brain, and there's so many positive things likely emanating from that. But I'm going to focus on muscle, and I'm going to start first here with some data on keto adaptation and glycogen metabolism. And showing you a graph here from a study we published back, I believe it was 2015, in elite group of ultra-endurance athletes that were either following a traditional high-carbohydrate or a traditional low-carbohydrate diet. I'm not going to go through all the data in that paper, but the low-carb athletes were prodigious fat burners. They had twice the level of fat oxidation and performed a 3-hour bout of exercise using about 90% fat compared to the high-carb athletes, which were using more along the lines of 40 or 50% fat. But what was absolutely astonishing to me was that their resting glycogen, their post-exercise glycogen, and their recovery glycogen levels were almost exactly the same. And you're seeing the individual responses here, so there's also a remarkable homogeneity in the responses here. There's no real outliers. And then this is in the context of these low-carb athletes eating a very low amount of carbohydrate. So quite unexpected, because I think we're taught that to maintain optimal glycogen levels, you need to be consuming a lot of exogenous carbohydrate. So how could this happen? So first of all, if you look at the rate of glycogen breakdown and calculate that out, again, it's nearly identical. No difference at all between low carb and high carb in terms of glycogen utilized per unit time. And if you do the math here, there's something quite odd. So if you kind of make a few assumptions here in terms of the amount of active tissue involved, you can calculate it in actual gram amounts how much glycogen was utilized. So based on these calculations, about 160 grams of glycogen was depleted during exercise. But we actually have indirect calorimetry data on the total amount of glucose oxidized that actually was terminally oxidized to generate ATP, and that was only around 60, a little over 60 grams. So the big question is, what the heck happened to 100 grams of glycogen if it can't be accounted for in breath and glucose oxidation? And I'm not sure I have the answer. It really has not been followed up on. But if we take a look here, drill down a little deeper into the biochemistry, so you've got glycogen broken down into glucose, and then, of course, glycolysis. And typically, if you look at the end product of glycolysis, pyruvate, a lot of that is being converted to acetyl-CoA through pyruvate dehydrogenase, and that's the entry point into the Krebs cycle for terminal oxidation. And of course, fatty acids can also serve as a source of acetyl-CoA. But we know that ketogenic diets actually decrease PDH activity, and most of the acetyl-CoA that's being used to generate ATP is coming from fatty acids, because you have an abundance of fatty acids when you're keto-adapted. So that's why I kind of have that redded out. It's unlikely a lot of pyruvate is going in that direction. But pyruvate can also, it has other fates, so it can be converted into oxaloacetate. And this is interesting because oxaloacetate is necessary. It's kind of an anaphylactic substrate for the Krebs cycle. You need oxaloacetate for the Krebs cycle to run. In fact, I don't know if it's still in the textbooks or not, but I was taught that fat burns in the flame of carbohydrate, which doesn't make a whole lot of sense when you think about it. But the basis of that was that if you're burning fat, you have to have a source of oxaloacetate, and typically oxaloacetate is derived from glucose, so that's where that comes from, even though it doesn't make a lot of sense. But that's likely perhaps one reason why in a keto-adapted state you need to have a fully functioning Krebs cycle, that perhaps breaking down glycogen and going through glycolysis helps keep the Krebs cycle running by providing a source of oxaloacetate. Pyruvate could also be converted to lactate, and that also, I have that green, because I think that likely occurs to a greater extent in these keto-adapted athletes. And lactate is a great source of carbon for the cell, and can even be used directly as an energy source. And then you also have this sort of other pathway parallel to glycolysis, the pentose phosphate pathway, and that produces some important products as well, like reducing equivalents, NADPH, as well as 5-carbon sugars that are necessary for nucleotide synthesis, for RNA, DNA repair. So there could be advantages to having a source of glucose in the keto-adapted state in terms of promoting reducing power and 5-carbon sugars as well. So I think this is all hypothetical, but all reasons why, from an evolutionary perspective, being keto-adapted, you still might want to have normal glycogen levels, typical glycogen levels. Now, what about the resynthesis of glycogen? So when this is looking at the post-exercise and then the 2-hour recovery time point, and the low-carb athletes here did not get any carbohydrate during this time period, whereas the high-carb athletes did. And still, they synthesized glycogen over 2 hours at the same rate. So it's kind of slightly different phenomenon here, but it's still not expected, right? I mean, if you're eating carbohydrate, you would think there'd be a higher rate of resynthesis. So how can you synthesize glycogen without any exogenous carbohydrate intake? And so the question is, can there be glycogen repletion in the absence of any carbohydrate intake? And typically, I think a lot of us would have said no, but if you look in the literature, especially across different species, the answer is absolutely yes. There's a variety of different animal models that have demonstrated glycogen synthesis after some type of physical demand and shown glycogen synthesis. The question is, how does it happen? What are the carbon sources for glycogen if it's not exogenous glucose? Well, it could be lactate. Lactate can be recycled back to the liver and converted to glucose. Amino acids, some are gluconeogenic and can be used as a source of glucose. Glycerol as well, and there's a lot of glycerol being released from adipose tissue in the keto-adapted state. So this is just showing, glycogen, if it's being broken down into glucose and converted to lactate within skeletal muscle, that could be released and delivered to the liver, converted to glucose, and then the glucose could be used as a substrate to form glycogen. So it's likely that process is upregulated in the keto-adapted state. And interestingly, the transporters involved, the MCTs, the monocarboxylic transporters, don't distinguish between ketones and lactate. they're both organic acids and use the same transporter. So you'd expect they're upregulated in the keto-adapted state. I don't know anyone who's actually measured these in a human or an animal study in this context, though. So again, hypothetical. And interestingly, if you look at the actual circulating substrates during recovery, lactate and glycerol were actually higher in the low-carb athletes post-exercise, and then you see a dramatic decrease in those substrates over that 2-hour recovery period. So where are they going to account for that pretty dramatic lowering of blood concentrations? Again, perhaps into glycogen. Again, those carbons clearly could be recycled into glycogen. Now, I don't know anyone who's followed up on that data in any human study, which is a little surprising. We should have probably done that by now, but anyone looking for a good master's thesis or doctoral dissertation, that's, I think, really needs to be validated. There is some animal studies, though, that are consistent with what we showed, and this is using a mouse model, a swimming model. So they had mice treading water for 60 minutes, and it's a model of glycogen breakdown and fatigue. And then they extracted muscle out of the mice and exposed them ex vivo to glucose and insulin and different levels of ketones. So I think they had one with no ketones and then 1, 2, and 4 millimolar of ketones and measured glycogen levels. And this is just demonstrating the model. So the 60-minute swim model did decrease glycogen, as you can see in the panel A. Panel B shows with no ketones, you're not getting any resynthesis. Or I guess that's 1 millimolar ketone, excuse me, no real difference. But with 2 and 4 millimolar, you're getting a progressive increase in glycogen synthesis over a 2-hour incubation period. So evidence, again, that ketones might have a role in restoring glycogen from exercise. And this has to do with the question of, can skeletal muscle produce ketones? So traditionally, we think ketone production or synthesis occurs in the liver, maybe to a smaller extent in the kidneys, but generally not to an appreciable amount in other tissues. So if you look at some of the enzymes in ketogenesis, they're very low in skeletal muscle, so SCOT being one of them. And that kind of makes sense because skeletal muscle has a lot of different ways to utilize fatty acids. There's a range of different transporters to get fatty acids into skeletal muscle, so there's not a real need to use ketones like there is for the brain. So it makes sense the body would prioritize ketones for the brain and other tissues maybe, but not skeletal muscle. But very few people have done human work in a keto-adapted state and measured any kind of muscle adaptations. So this was one study we did that involved taking muscle biopsies out of individuals that were fed a ketogenic diet for 12 weeks and trained as well during that time period. And we had a control group that consumed a traditional high-carb diet. And we did demonstrate, my doctoral student at the time, Vin Miller, isolated mitochondria from those skeletal muscle and showed increased mitochondrial protein content and an enhanced ability of the mitochondria to utilize fat in particular. But interestingly, he also looked at ketone oxidation in those mitochondrial isolates and showed very, very low levels. So that's kind of consistent with this idea that muscle's probably not producing or utilizing ketones to any great extent. Not that it's zero, but a pretty low extent. And then this is also data from the FASTER study, so the ultra-endurance athletes that were low-carb or high-carb. So we did, from muscle biopsies in that study at 3 different time points, we looked at transcriptome analysis. So we looked at gene expression of all known genes. I think we had quantitative data on over 23,000 genes at each time point. And of all of those data, the gene that had the most significant difference between the low carb and the high carb group was HMG-CoA synthase, which is codes for the rate limiting enzyme in ketogenesis. So that is not happened by chance. There's something, for some reason, this particular enzyme is increased in athletes who are keto adapted, and it occurs pre-exercise and post-exercise. So I don't know why this would go up if you're not producing ketones in skeletal muscle, but the gene expression is highly expressed in these keto adapted athletes. Now, we know that ketones can actually be produced from leucine through an alternative breakdown or pathway of leucine degradation. And people have studied this as well, mostly animal models though. And it's a pretty low percentage of the total ketones, but it's been estimated in animal studies that about 4% of total ketone production is from leucine breakdown. And that probably decreases as you become keto adapted because of the leucine sparing effect. But nonetheless, in a truest sense, ketone production does occur in muscle from leucine to some extent. And what's even more interesting here, and this is a little nuance, but it's not evidence that ketones are produced in muscle, but there is some transformation going on within skeletal muscle. So there's been about a half a dozen studies published now, and most of these use the forearm arterial-venous difference model. So they'll literally catheterize an artery and a vein that feed and drain the forearm muscle in people, and look at the delivery of substrates, in this case ketones, and then what comes out on the venous side. And what you see is, so if you focus on the first two rows there, those are the ketone levels that are in the the arterial side. So you've got ketone levels over 60 hours or so of fasting, and you see those are micromolar, but less than 0.1 millimolar, that then they increase to almost 0.5 and for acetoacetate, and then almost 1 millimolar, and then beta-hydroxybutate's quite a bit higher. Now the two rows below there, that's the flux, and you see the flux of acetoacetate is much higher, and beta-hydroxybutate's actually negative. So what's happening here is there's a dramatic increase in acetoacetate uptake into skeletal muscle as you become keto adapted, or during, in this case, prolonged fasting. But what comes out the other side is beta-hydroxybutyrate. So there's a conversion happening within skeletal muscle. And of course, that enzyme is beta-hydroxydehydrogenase. So there's uptake of acetoacetate, but then a release of beta-hydroxybutyrate. And this is almost assuredly the reason why we always have more beta-hydroxybutyrate in the circulation than we do acetoacetate. Usually, it's around 60, 70, 80% of the total ketones. It can vary depending on the condition, but what's coming out of the liver is not that ratio. It's more of a 50-50 ratio. So this peripheral conversion of acetoacetate to BHB is happening specifically in skeletal muscle. Now, you could say, well, so what? Maybe that's because the brain prefers beta-hydroxybutyrate. I don't think that's the case. I've asked a lot of experts. The brain doesn't care if it's acetoacetate or beta-hydroxybutyrate. In fact, you got to convert it back to acetoacetate to oxidize it anyway. But if you look at that reaction, beta-hydroxy dehydrogenase, look at the cofactors, it's one of these redox reactions, right? It produces NAD+. So being keto adapted and undergoing this transformation provides a source of NAD+ for muscle cells. And if you haven't followed that literature at all, NAD+ is super hot molecule right now. It's being linked to all sorts of positive cellular functions within cells, particularly related to longevity and so forth. It's just one of many recent reviews out on NAD. So here we have a ketogenic diet, probably you could say is a really good way to keep adequate stores of NADA, because we know those go down, for example, in aging. Okay, third aspect, ketones and heart function. So just like the brain utilizes ketones in proportion to the delivery, so there's a really tight correlation between uptake of ketones and the circulating level. I think Stephen Ketone has shown that many times in some really beautiful graphs. But that same relationship is true for the heart. So as ketones are elevated in the circulation, the heart takes them up and utilizes them in proportion to the circulation. That's certainly not the case with skeletal muscle. There's the uptake is, there's a lower Km for the transporter, and you saturate that process at a much lower level. But with heart, the higher they go, the more the heart utilizes ketones. And a lot of new data coming out now showing that when you have a ketone-fueled heart, it's functioning more efficiently and better, particularly in disease states like heart failure. And so in the context of heart failure, there's all this data coming from a variety of different sources indicating a ketone-fueled heart is functioning better. Now, some of this is just from the metabolic side of things, is like I just said, the heart does utilize ketones if they're available. It's an organ that needs a lot of energy, and it needs it on a regular basis to function. So it will tend to oxidize anything that's there, but it will preferentially use ketones. There's also clearly a metabolic basis to heart failure. So heart failure is associated with impaired ability to utilize substrates, in particular fatty acids, and an increased reliance on glucose. And so there's a lot of different animal models out there now, and even human studies showing that the failing heart will utilize ketones to a greater extent. And then you've got animal studies. I think Veach published one of the first many years ago showing that a ketone-fueled heart in an animal model functioned more efficiently, so it produced greater cardiac function per unit ATP when it's run on ketones versus other substrates. But even in the last, say, 5 years or so, now we've actually got human studies in both clinical populations as well as healthy populations showing that when you elevate ketones acutely, you get beneficial effects. And I'll go through a few of these in a minute, but we've got ketone infusion studies. And more recently, in the last couple of years, we've got a couple now papers out on exogenous ketones, elevating ketones acutely and improving cardiac function. And then the last bullet point there I'll just mention, this is kind of another way in which I think cardiologists are starting to come on board with this. We have quite a lot of clinical trials now on SGLT2 inhibitors, which are used to lower glucose in diabetes. But we have findings now published showing improved heart outcomes in diabetic patients and even non-diabetic patients now with heart failure, showing reduced hospitalizations, decreased cardiac events with just use of these drugs. And these drugs cause increased glucose output in the urine, but one of the other metabolic effects of these drugs is they elevate ketones modestly. So typically up into the lower end of nutritional ketosis, so maybe 0.3 to 0.5 millimolar, without following a ketogenic diet. But a lot of people have speculated that the cardioprotective effects of these drugs might in part be due to the ketones. So that's just another level of evidence supporting ketones having a beneficial effect on heart function. One of the first studies, this was the ketone infusion study out of Sweden, where they looked at patients with heart failure, reduced ejection fraction, and infused a ketone salt, a beta-hydroxybutyrate salt, into these patients. And you can see rapid elevation in their circulating ketone levels. And amazingly, when they looked at cardiac function, then you get this dramatic increase in cardiac output. So, and it's kind of a dose response. So you can see the upper right bar graph there showing increase in cardiac output as a function of elevation in circulating ketones. So the greatest effect was at, is that 3.3? I can't see. But increasing cardiac output with higher levels of ketones, greater increase in ejection fraction, and there's a vasodilatory effect as well that has been observed in most of these studies. And another interesting part of this study is they did the same experiments in healthy controls with no heart failure and showed the same effect. So this wasn't just working in people with a defective or impaired heart function. It actually is a very robust effect that occurs across the spectrum from health to diseased hearts. So we now have a couple studies looking at exogenous ketones as the method of elevating ketones, and one was, this is in healthy participants using the ketone ester, or the product from human. You'll notice increases in ketones are very similar to the ketone infusion study I just showed you, so a little over 3 millimolar. And this is showing the improvement in cardiac output with that acute elevation from ketone supplements. And then this is even more fresh off the press. I think this was published just in last December. Again, 20 healthy adults. This particular supplement was Ketone Aid. And again, ketones, I guess, maybe not quite as high here. They got a little over 2 millimolar with a single dose of this particular exogenous ketone. And then you can see it on the right, cardiac output progressively increasing over time. And then, of course, if you look at cardiac output as a product of stroke volume and heart rate, you see it's contributed, both are contributing to this increase in cardiac output. In this particular study, the heart rate is actually driving more the increase in cardiac output than the stroke volume. We did kind of a pilot preliminary study similar to the two previous projects I just showed you, and we're studying the ketone ester from Juvenescence, which is this was the older version, the C6 diester, and again, single dose delivery of this particular product to healthy subjects, and then serial imaging and blood draws after that. So we've just finished this study. We're close to getting this packaged up for submission. So these were presented last year at an imaging conference, but otherwise unpublished. So we get a nice increase in ketones, which is no big surprise. It takes a little longer for this C6 diester to elevate ketones, so it's a little more gradual, but by 2 hours we're at over 2 millimolar, and get a little bit of a glucose lowering effect with that. But the cardiac output data are very similar to those previous infusion and exogenous ketone studies, so we're seeing a highly significant increase in cardiac output. And we did use a placebo as well, a calorically matched placebo, fat-based placebo, and we didn't see any increase. So this was kind of one unique aspect that we added in addition to the advanced imaging. So this wasn't just an effect of food intake and caloric intake that people have seen. Now, if you look at the heart rate and stroke volume aspects of the cardiac output, again, it's both are increasing, so there is a sort of a chronotropic and inotropic effect both going on here, increasing as a function of elevated ketones. And then we also do a measure of myocardial blood flow, and that increase similar to what's been shown in the infusion studies.